few things about a very new field. This field is the analysis of ancient DNA and what we can dig out from there. And we can dig out a lot of things. We received this chance in the last few years because the technologies, about 5 years maybe ago, wasn't ready for it. So that's not as much as what we have already dug out, but rather something that we want to go into. There are many studies which we try to understand, in which we try to understand whether humans invented fire. How did it influence humans? Food was raw and running before that, and now it smells nice and it's hot. It should have some influence. There are many articles which were sometimes written many years ago where people were trying to work out how cooked food influences the microbiome, the genes that influence the selection, and so on. And what happened? So the best way to understand how it influenced humans is looking at certain anthropological data. So we dig out an ape, an extinct ape, and we're looking at what happened. What is its stomach volume, brain volume, and we compare that with anthropological changes. That will be secondary data because we won't know what happens in the genes. To understand what happens in the genes, we need to sequence this ancient ape or Australopithecine or Pithecanthropus. There are difficulties about that. One of the motivational articles that made me interested in this topic was an article that outlined the interest of public for this subject. When people started cooking, the energy got bigger, larger, and various parasites, worms were killed. Many bacteria were killed during the cooking process. So humans could now have a smaller stomach and a smaller gut. They didn't need huge molars like the Little Red Riding Hood's grandmother. But at the same time, when humans were cooking, certain things are burned, and there are toxins that are connected with cooking. The individuals who didn't have any protective reactions against these toxins, they were weeded out because they were diuretic all the time. What is very interesting is how various remains, ancient human remains, were excavated. The time when humans invented fire and the time when the first changes were found, these times do not coincide. Why? It became clear that evolution is not a momentary event. Other time and many generations must pass before cooking and the smaller teeth. So this gap is rather big. Just time-wise, the mutation appears much later. There are some cases when we see the opposite. A mutation that makes it possible to digest cooked food appears before the first evidence of cooking. Well, perhaps we haven't found all ancient fires. Perhaps our archaeological record is incomplete. But perhaps there was some chance mutation and it became difficult for humans to digest raw food. So they invented cooking. It's not clear which came first. There is a group of genes, which is like an abbreviation from taste, TAS. Many genes that are involved in metabolic pathways of digestion and taste reactions. Many genes from that pathway, if we compare ancient humans and modern humans, many genes have changed. Again, it's difficult to say anything for certain because we don't have enough ancient genomes sequenced. But the mutation in those taste genes, as compared with other primates, or Neanderthals and Denisovans, they are different. So cooking makes people appreciate taste differently. For example, we love the smell of smoked sausage, which perhaps wasn't like that for ancient humans. Another thing that happened is with agriculture, with the advent of agriculture, when humans started cultivating wheat, rice, they developed a different reaction to starch. A wolf will not be able to digest that, but humans are. So how do we make sense of it? Of course, we can hypothesize about the beginnings of agriculture, the beginning of the invention of fire, and now let's look at ancient genomes. How can we find traces of selection and have a look at what was happening there? Very recently, the next generation sequencing. Have you heard of it? When we can sequence small parts of DNA, which is very cheap and very fast. Primary Sanger sequencing was quite difficult, but now we have a chance. And that's why every month now we have sequencing of an ancient mammoth or an ancient horse, and comparing those genomes with modern animals, we can find many interesting things about evolution. We wrote a review with one of the ladies who's here. We wrote a review about ancient DNA and what is the current situation with that. It will be published sometime around this month, and we wrote the most interesting things about that. What is very important about that is when you analyze ancient DNA, you have to compare anthropological data with sequencing data, and I'll say a few words about why it's important. First, you have to separate the chaff from the wheat, because ancient DNA is very contaminated, and you have to formulate archaeological hypotheses and then check them and verify them using sequencing. There are several ancient hominids, humans, that we know of. These are Neanderthals and Denisovans. As you know, Neanderthals and Denisovans were one of the first humans who came out of Africa, and when the ancestor of the modern humans came out of Africa next, crying, "Hey guys, wait for me!" Those were already adapted for life in Europe, and then transferred many protective genes that they have developed in northern climates. For example, better cold resistance, lighter color, lighter eyes. We received that from Neanderthals. As for Denisovans, they went east. And Polynesians, they have a larger trace of Denisovans, not Neanderthals, because this is the group of our evolutionary cousins who went to the right. It wasn't like an evolutionary tree, but these humans were working to and fro. There were no borders, no passports, so people were really walking a lot, as much as they could. So it's very difficult to say who is whose direct ancestor. There are lots of hypotheses, some of them are conflicting between each other, and DNA that we find is fragmented. Depending on skeleton, the percentage of ancient DNA that we're interested in is from 1 to 10 to 20%. So that's all the genome that we have covered. we say that these genes were received from those hominids. Perhaps that's not true, because we have an experiment based on one sample. If you know statistics, when we have one sample, we can build any theory upon that. And then we'll have another sample, and we'll say something else. I'll show you such examples. Another misconception: why did Neanderthals go extinct? What have you heard about that? What did you heard about Neanderthal extinction? They were eaten by anatomically modern humans. Anything else? There was a hypothesis that they could not talk efficiently, that our ancestors could chat, were still chatting quite efficiently, and Neanderthals couldn't. That's why when the first sequencing of Neanderthal genome was performed, they didn't find the speech gene. But in later research, which was sequenced much better, they found the FOX2P gene in Neanderthals, and this gene is known for a mutation which makes humans unable to speak. But the Neanderthals had a functional gene, and it seems that they could talk. Perhaps there was some other reason why they went extinct. Another interesting thing that we can understand using the analysis of ancient DNA is comparing the genomes before the Bronze Age and after the Bronze Age. As you know, when our ancestors domesticated goats, cows, and other animals, first they started getting dairy products, then drinking milk, and so those who could not digest lactose, they had diarrhea and they couldn't digest it, and they also got weeded out of the gene pool. They domesticated cows about 10,000 years ago. The gene responsible for, or rather the frequency of that allele, it appeared something like 5,000 years ago. So for 5,000 years, man was living next to a cow and couldn't drink milk. They probably ate cheeses, or maybe they used cows for some other purposes, but they couldn't drink milk. And you see the comparison here, which shows when the allele appears that enables the digestion of lactose. Obviously, what happened wasn't sudden. For a new version of a gene to appear, it has to afford some evolutionary advantage. Of course, somebody who can digest milk can grow faster and have better bones and so on and so forth. Could have more wives and so on. But about 8,000 years had to pass before that came about. Dima Alexeyev is here, and maybe he can add something to that. But what happened with the development of technology, with the industrial processing of food? It led to the change in the oral bacteria. It was happening in the 19th century. As soon as we had a gene enabling us to digest cooked food, the oral flora also changed, and the bacteria that used to be very diverse with our ancestors. And we can see that because the tartar, it preserves bacteria very well, and sequence that it's like a time capsule. And we can have a look at the diversity of bacteria in our ancestors. And here you see modern tartar, and there is time lapses before that. They are clusterized in different parts regarding the variety. And another thing, we didn't just lose diversity. Our ancestors didn't have bacteria that were carcinogenic. Or perhaps they had, but not so many. Those of you who have donks, you know that donks very rarely have caries. And we have it all the time. I don't have a single tooth without a cavity. Well, I'm unlucky that way. But this is because modern humans have fewer bacteria, and those that we have cause caries. This is the price we had to pay for industrialization. What we eat influenced the development of bacteria in the mouth. And this is a very interesting riddle from the Huesos cave in Spain. How interpretation of incomplete data leads to silly hypotheses. There was a skeleton from the Los Huesos cave, and when they looked at the teeth looked like a Neanderthal. Then it was sequenced, a small part of DNA that they could sequence mitochondrial DNA. They didn't have enough money, so what they had, they sequenced. That was a human who lived about 50,000 years ago in Spain. But his mitochondrial DNA looked like a Denisovan. So the Denisovan went to Asia, and we're finding in the far west of Europe a Denisovan type DNA. Nobody understood what that was. But then, when we got more money and the technology got better, they sequenced most of the genome, and it was a Neanderthal after all. And it was a chance that they had a Denisovan-like mitochondrial DNA. We understand that they are heteroplasmic DNA, and we have a chance that they sequenced an atypical molecule. And this small sample made them conclude some silly things. And this is a danger of working with ancient DNA, because you take a piece of DNA and you start saying, well, they went to the right, they didn't went to the left, didn't go to the left, and so on and so forth, which is wrong. This is the size of the molar. These are the teeth. These are Neanderthals, the men from Cima de los Huesos, and they are very much like Neanderthal. And Denisovan molars are huge. With ancient DNA, you have to be very, very careful. And the sensational news about Denisovans living in Spain, you have to really be careful about that. Now, let's have a look what is already excavated and sequenced. This is a map from our article where we are showing various European and other locations. Why do you think there are many locations in Europe and not too many outside of Europe? Well, they have more money and more interest in Europe. It's the same in America. They can't dig or excavate in America because there was a very stupid case when an Indian boy, a Native American boy, was excavated, and an article was published without the permission of the tribe, and the migration way was different from what the tribe believed, and the Native Americans were offended, and this is an ethical problem because now they forbid publications, and you have a 5,000-year skeleton. It doesn't really matter. No legends are connected with that. If we're speaking about some more recent past, you can offend someone. You remember the Romanovs scandal. Is Anastasia alive or not alive, and so on and so forth. This is a very emotional thing. The closer it is, the more emotional it is. So Europe is covered reasonably well, and here we can see some Denisovan samples, and here that's mostly Neanderthal, Bronze Age. Europe can be studied pretty well. And have a look at our contribution to ancient people. What is the difference between working with modern and ancient DNA? First of all, ancient DNA is fragmented. When people die, his or her molecules start to decompose. It's contaminated. Then there are wolves who add something, so there are some wolf bacteria. There are chemical modifications, which just happens. Cytosine becomes uracil. And so there are time frames. If anything is older than 1 million years old, it won't happen. You can't sequence a 5 million years old remains. 560,000 years ago, right now, an extinct horse was sequenced, but that's the oldest we can get. The longer time has passed since death, the larger percent of bacterial genomes is in the DNA. Then contamination again. That's why there were a few silly cases which you should be aware of when a dinosaur DNA was received, but it wasn't. It looked very much like modern humans. That was a false sensation because that was from a lab assistant, of course, and not from a dinosaur. Another curious case was when they were looking for a salt crystal, looking at what bacteria remain there, but the crystal was very old, and the bacteria was modern E. coli. Somebody didn't wash his or her hands. So how can we fight contamination? You must work in a clean room, in almost a spacesuit. Then, of course, you have to take off the outer layer of the bone, to bleach it, to remove the layer that is susceptible to contamination. There are difficult cases. For example, you're digging the graves of the Napoleonic War, and you are looking at Russians who lived 200 years ago. What's the problem? If you are the same Russians who are working with our great-great-grandfathers, your DNA will be processed much more joyfully because it's younger, and it will be difficult with their DNA because it's fragmented. It's not that good. And you will be sequencing yourself, not your great-grandfather. How can you tell one from the other? To work with European great-grandfather, you have to take a Chinese lab assistant or an African lab assistant, because in that case, you would be able to separate certain mutations that are typical of the Chinese or Africans, but doesn't exist in Europe. There is a different approach, but it's not a good approach. If you receive a long DNA chain, it's likely to be modern. You have to throw it out. But the decomposition of DNA is stochastic, but sometimes, by chance, you're very lucky and you receive a long molecule, maybe like with the pterygian mammoth, you can have something very good, a very long chain. But this is dangerous, of course. Or if you were to work with mammoth, you take a mammoth and you need to compare it with an elephant, a modern elephant, because an elephant should be more or less like an elephant, and there is unlikely to be any contamination from elephants, right? So for different time periods, you have to choose different bones. For example, temple bone is very good. For some other times, hair is good. But it depends upon which periods you're working with. So there is no single answer, and different scientists will be telling you that you have to take this. You have to be looking for errors, because in ancient DNA there is deamination of cytosine. So C goes to T, and if you have some reference genome, you have to have a look at how many of such changes you have. If there are no changes, you have contaminated modern DNA. If you have such changes, then it's correct. You have to be very careful without mathematical methods. Depending on the quantity of errors accumulated in ancient DNA and the difference of an ancient genome from modern genome, different methods will be working differently. If you work with a modern DNA, almost all alignments will work well, but with ancient DNA, not so much. So this shows how many thousand years ago the separation was, and which methods will work or won't work. So the selection of tissues and meta-analysis, it depends very well in different cases. There is a notion of the mustache of antiquity. At the ends of a molecule, you're seeing some kind of a strands, overhangs, and if the molecule is still rather tight, it doesn't happen that much. So you have those overhangs of antiquity. The errors accumulate at the ends, and this is what you're looking for in ancient DNA. If you have uracil, what do you do with that? It can be the former T, or it can be an error. So again, it's not very clear what to do with that. You can look at the distance from the end, or you can take multiple samples and look, because they happen by chance. So statistics is the only way to work with that. Well, again, this is Ancha's diagram. What should you do? How can you check your sample? Okay, you check this, you check that, and so on, and then you proceed. If you just take the modern pipeline and use it to sequence a Mammoth or a Neanderthal, you won't get any sensible results. You have to use new methods, new probability-based methods, because this is a probability-based analysis. And if you want to make any conclusions, to draw any conclusions, for example, about the diet of the ancient humans and the influence of the diet on DNA, but you can't separate errors from mutations. So your conclusions should be based on groups of genes, not single genes. And this analysis should use additional data. We wrote a small algorithm which uses ancient genomes with certain data, and we looked at who looks like who. I'm proud of this arrow here, which goes from Europe to Britain. You can explain that. What do you think about that? What is that? Bronze Age. Who went from Central Europe to Britain? The Celts. The Celts. They just left and went there. And these are German tribes who proliferated there. Maybe Attila was going here and there, to and fro. So it explains it very well. Coming back to the importance of mutation, 23andMe is doing something that I don't like. They're looking at one SNP and they say you have this mutation, that means you love coffee. You have this mutation, that means that you have blue eyes. For some things it works. I was looking at my own profile in 23andMe. Half of what they said are just nonsense. They told me that I was a light-headed blonde woman. Well, I'm not. It should be more. It should be a more detailed approach. We're taking mutations which we compare with a certain metabolic pathway, and we're looking at what phenotypical changes occurred. Now, just a few words about my postdoc, who did a few very interesting things. We don't always have ancient bones, but sometimes we have some isolated populations where we also can find something. Where are we going to be looking for them? In Africa. Well, because this is the cradle of civilization, there are many isolated tribes. They have unusual languages, the largest language diversity of the world. And you can be looking at different groups who eat different things, different diets, and have different lifestyles, and to look at their difference from each other. You can get 100 or 200 genomes there because they are there. So this migration corridor, this is something. This is where people were going from Middle East to Africa and back. And you see here that many things were changing. Sometimes there were droughts. Sometimes there were more humid periods and cooler and warmer periods and so on. Many things were changing. So there were many groups that began as a single nation. Some other became sedentary. Some other became nomadic. And the divergence is here very important. But I have nothing to do with this work, with this article, but I invited this postdoc to join my team. Fulani is a very interesting nation, very beautiful. So one third of them are nomads, and 2/3 are sedentary. According to a legend, they came from someplace in Egypt, and then they settled in West Africa. They have a major European admixture. So Peter developed mapping mutation onto metabolic pathways, and they were trying to find the genes responsible for lifestyle adaptations. They found in one of the subgroups a very strong taste receptor signal, just in one group, in Fulani group. And when they looked at that was on chromosome which had a major European admixture. So something changed, and they had a major signal in taste receptors. The only thing which was different from the other people, they have a very interesting custom when they become adults, an initiation ritual. They are flagellated by the whole tribe. This is a very cruel ritual. If a boy cries, he doesn't become a man, he has to leave the tribe, and so on and so forth. How do they survive? They drink themselves silly. They make a special drink from Tatarinova. It's horrible, and they have to get used to that. So to drink that, they have to drink several years before, because it's very sour, and it's okay for them, because those boys who could not drink that, they were crying during the ceremony, and nobody married them. So that was a selection pressure based on taste receptors. Or Maasai, we accumulate fat on our buttocks. But these scoundrels, they can eat as much fat as they want. Their whole diet is cholesterol rich, but still they're very skinny. I just want to kill them. They're eating cholesterol more than European, like 6 times more than an average European. And sometime around 1971, I think it would not have been allowed now, they took healthy boys, aged young lads, 20 to 24 years old, and a similar group of Americans of mixed ancestry. They divided them into treatment and control. The control group was just eating a regular diet, and the treatment group was given 2 grams of cholesterol per day in addition to what they already had. So the Maasai were not affected. Heart, blood, nothing. Weight gain. The Americans gained 11.8 milligrams of cholesterol was a raise in Americans, and Europeans and Americans. In them, it leads to various cardiovascular diseases, to many troubles apart from just aesthetic troubles. But with Maasai, they don't have any coronary disease at all. What's interesting about that is that this is some kind of a defensive mechanism. They used to live with subsist on a very fatty diet because of their agriculture. If you're eating that quantities of fat, you wouldn't survive otherwise. So they loved to eat fat, lard, and those who couldn't survive that, they just died. And it's an interesting society. They are polygamous, and adult males who are 70, 80 years old, it's good for them to marry very young girls. And for that, they need to have healthy heart, because otherwise, given the amount of fat they receive, they wouldn't be able to procreate. But this kind of sexual selection, it's very necessary for old men marry young girls, and that put an additional sexual selection pressure on them. For us, as Europeans, it's probably not very helpful. But it's an interesting fact. If there are certain mutations in certain genes, they can allow people to go to McDonald's and stay healthy and have a tiny waist and a good heart. But if you are not born that way, too bad. A mutation or a group of mutation can make some people digest various bad things. It probably means that we can create drugs that would increase the activity of those genes, maybe some gene therapy, then maybe you can eat lard using these tablets. This is the beginning of research, and this research is only possible because we can now use ancient DNA that I talked about earlier, and now sequencing is so cheap and so easy that we can look at large cohorts, that we can accumulate statistical values, and we see the difference in genomes in people who can eat carrot or can't eat carrot. Right now, it's very easy to do. And we're just at the beginning of this road, but this is extremely important, not just for nutrigenomics, but for pharmaceutical genomics. Yesterday, I heard a report about the mutations that the WHO is recommending for testing of certain anti-tumor therapy. The Yakutians don't have those mutations. So something that we recommend for Yakutians, it won't be helpful for them, because these these drugs were made for Europeans, and we need to do something different from them. Of course, we are Homo sapiens, all of us, but for some of us, lard is okay, but for some other nations, it's not good. And I think this is very important for us. Thank you.